Hello, good morning, scientists, and you're welcome back to our science community. It's always beautiful to have you here as we continue, in gaining more understanding in the world of science and life sciences in particular. Today, we are talking about PCR. That's polymerase chain reaction, PCR. Now, this is a laboratory technique that is well used in the life sciences, and it's a laboratory technique that is used for many purposes. But the underlying principle, the most important thing in conducting a PCR, is trying to take a sample and get a sequence of a DNA, a sequence, and then amplify or multiply this sequence into thousands in such a way that this sequence now becomes more available, accessible, and can be studied, and is more important for use. Now, this technique has been around for quite a while and has been used extensively in the field of life sciences to try to understand quite a number of things. PCR, there are so many different types, and over the course of these videos, we'll discuss all the different types, real time PCR, quantitative PCR. There are so many different types, and we'll get to understanding them. But it's important to understand that what is the underlying principle? The underlying principle is this: when you have samples that are live samples. When I mean live samples, I mean samples that are either microorganisms, they are either eukaryotes or prokaryotic cells. Then you go into these cells, and these cells have DNA. And then this DNA, have genes, and these genes are in sequences. And many a times you want to study a particular gene or a particular sequence. And because usually DNA is in very small and minute quantity, and that makes even the sequence you want to study be in a much smaller quantity, then what do you want to do? You want to create, or we scientists created a method where you can take a small sample of DNA, and then you also have a target sequence that you are thinking of studying, and then you can make thousands of copies of that sequence that you want to study, make those copies, and then it is more amplified, and you're now able to study it better. You're able to understand the individual nucleotide sequences, the nucleotide that make up those sequences. This is very important in life sciences. And when we talk about PCR, there are three major methods or three major steps that go into the process of conducting a PCR, a polymerase chain reaction. And these processes are, you have denaturing, which is number one, you have annealing, and then you have extension. Just three major processes: denaturing, annealing, and then extension. Now, when you talk about denaturing, usually your DNA is a double-stranded structure. You have two strands, two complementary strands that make up your DNA. And so when you want to run a PCR, a polymerase chain reaction, what you initially need to do is to denature the DNA of interest. And denaturing in the sense means that this double-stranded structure, you want to unwind the strands such that you have single strands on both sides. This is the beginning process, and is very important. Now, when you denature the DNA, meaning that you separate the double-stranded structure into a single-stranded structure, then the next process is what we call annealing. Now, in PCR, we have what we refer to as primers, and primers are short sequences, or short nucleotide sequences, which can mimic our target area. So you have a sequence that you're interested in, you have primers that look, are similar, but shorter sequences that are similar to your sequence of interest. And so when you first do your denaturing, where you divide your double stranded structure into single stranded structures, and then from there you add your primers, and that is the next process, the process which we refer to as annealing. And in the process of annealing, you add your primers, so your primers are able to target particular points along the length of your DNA, because they mimic what you have on the DNA. And so when they are attached to those points on the DNA, then the third process is the process of extension. And the process of extension is now the process where you begin to, or you are the process, or you yourself as a scientist now starts making new strands from the template strand. What's the template strand? That's the initial strand that you got from your double helical structure. So your double stranded structure has been divided into single strands. Then one of those strands you use as your template or your target DNA, and then you add your primers, which attaches to sites along this single-stranded DNA, and then the process of extension, more nucleotides are now added, and then you now begin to make new copies of the template DNA. This is really important in life sciences, and we have a lot of usefulness of PCR. We'll go further when we do new videos this week, maybe next week. We'll spend it talking majorly about PCR and what it's really about, why it is useful in the life sciences, and for which purposes that we get to use PCR. Thank you for listening and follow us on this PCR journey. Remember to like, subscribe to our channel, and do have a beautiful day.